simplification. Actually, this is the approximation of the probability density function. We make a simplification by modeling the diffusion as a tensor. So this is the tensor model. As we can see, this is the rank 2 tensor. So if the 3 eigenvalues of the diffusion tensor are all equal, then you will imagine the diffusion is the same for every direction, right? Like this. But if the first eigenvalue of the diffusion tensor is greater than the second and third eigenvalues, we can imagine that the water molecule diffusion is like this. That means in this direction, the diffusion is highest, but in other direction, the diffusion is smaller, right? So this is the shape we can imagine. If the first and second diffusion eigenvalues are equal, and they are all greater than the third eigenvalue, we can imagine the diffusion is like this, right? The probability of the diffusion is like this. So this is an approximation of the diffusion. We use tensor, a rank 2 tensor, to model the diffusion. And this will actually make us very convenient. It will actually allow us to detect the direction of the highest diffusion using this diffusion tensor imaging, or simply called DTI. This technique can allow us to detect in any position of the brain what is the highest diffusion direction. So we can actually apply diffusion gradient in 6 directions, at least in 6 directions, okay? And then we can put into this equation, and we can solve this equation using some linear algebra. And then we can get the diffusion tensor. This is a symmetric diffusion tensor. So there are 6 independent variables. 6 independent variables. So that's the reason why you need to turn on at least 6 independent diffusion gradients to solve these 6 unknowns. So if we can apply a diffusion tensor imaging sequence by using these 6 diffusion gradients, we can solve for diffusion tensor at each position. So now we have the technique, DTI, to solve for diffusion tensor at each position in the brain. And tensor will tell us many information, many pieces of information. For instance, we can calculate some scalars. For instance, apparent diffusion coefficient, ADC. It is simply the average of the 3 eigenvalues, okay? And also we can calculate an index called fractional anisotropy, FA. FA is simply the variance of the eigenvalues divided by the norm of the eigenvalues. So this is a very simple concept. And this actually represents the mean diffusivity of that location, okay? The mean diffusivity of the water molecules at that location. And this simply represents the directional anisotropy of the water molecule in that at that position. So we have 2 very simple indexes, and this is the mapping of these 2 indexes. This is the ADC map. As you can see here, the brain, every pixel in this brain represents how the ADC is varying. For instance, the ventricle, because there are a lot of water, and in the surface of the brain there are also a lot of water. So the ADC is high, very high. And inside the brain, you can see the ADC is lower because there's less water than the pure water, because it's all brain tissue, right? So this is the ADC image. And how about FA image? more interesting finding we start to see. Here we can see that these bright regions are all in the white matter. We have white matter and gray matter in the brain. White matter actually is the place where there are all axons. Axons, they gathered in the white matter. So here are the white matter. And this is the gray matter. Gray matter actually is the place where all the neurons or glial cells, they aggregate in the gray matter. So here is the gray matter. And also inside the brain, there are some gray matter. So we can see a very clear distinction between gray matter and white matter in terms of FA image. In the white matter, FA is higher than the FA in the gray matter. So we can see this kind of a very beautiful image. Why the white matter has lower FA than the gray matter? Why? Because in the white matter, because they are all axons. And axons actually is a restrictive environment for the water molecules. So water molecules in the white matter cannot diffuse very freely, okay? But in the gray matter, because they are all cells, cells are large and the water can go inside and outside of the cells. So water molecule has more free diffusion in the gray matter. So that and because higher restriction in the white matter, so the white matter actually shows higher FA because there is a directional dependence of the diffusion in the white matter because they are all axons. Okay, and gray matter there are all neurons, and neurons have no spatial directional dependency. And this is the color map of the FA image. And this color means the direction of the first eigenvector. So we can see, we can use the direction of the first eigenvector to represent what the axonal direction is at each position. So here we have red color. Red color means going perpendicular to the screen. And this blue color means they're going left and right direction. And green color means they are going up and down direction. So we use color coding to represent the direction of the neural fiber direction at each position. So here we have this kind of information.
So what's the role of blood vessel here? I mean, blood vessel is also a restrictive tube for water.
Yes.
Right? So will the signal mix with blood vessel?
Yeah. In the diffusion sequence, actually the signals from the blood or inside the blood vessel are all gone. Yeah, are all gone. Because the signal inside the blood vessels, actually there is flow in the blood vessels. And velocity will exaggerate the signal attenuation. So actually signal will be all gone. So there's no signal, virtually no signal from the blood vessels. Right. So here we can see this is the conventional MRI, and this is the gray matter, this is the white matter. This is the white matter, this is the gray matter, okay? And you can see if we just plot the first eigenvector of the diffusion tensor, we can see these lines, right? These lines represent the direction of the neural fibers at each position. And we can start to see, there's some patterns that we expect to be reasonable, because we expect the fibers should go like this way, and here the fiber should go this way. So it starts to provide us another new information, which is the fiber direction. We can detect fiber direction in the brain using diffusion MRI. So now I want to say something about some applications. Here we use diffusion tensor imaging to detect the prostate cancer. This is the prostate, okay? This is the urethra, and this is the ejaculatory tube. So this is the prostate. So we scan the prostate in this direction. So here we can see the prostate, okay? This is the prostate. And here I highlight as a red and yellow region. This region actually shows decreased diffusion coefficient, okay? So what does that mean? These regions actually are the cancer regions. So this is the prostate cancer in this region. And why the diffusion coefficient will be decreased in cancer? Because water molecules in the cancer, because there are many cells aggregate in the cancer tissue. And so most of the water are inside the cancer cells. So the water inside the cancer cells, so the diffusion coefficient will be decreased. So that means as compared to the normal tissue, the cancer tissue will show decreased diffusion coefficient. And we do the correlation between the ADC of the cancer tissue and the Gleason score. Gleason score is a pathological scoring system. If it ranges from 1 to 9, 1 means actually very benign, and 9 means the cancer cells are very aggressive. From microscopy, you can see the nuclei are very large, and the cancer cells are very bizarre looking. And this is the grade 9. So we have Gleason score to categorize how advanced this cancer is, okay, using the Gleason score. This is the pathology, okay? And we found that the ADC actually shows a very significant correlation with the Gleason score. If the ADC is lower, the Gleason score is higher. That means if you have a very low ADC, like 0.75, then your Gleason score is probably around 9 or 8. So this is that means you have poor prognosis, because Gleason score can predict your outcome in the later after the treatment. So if you have higher Gleason score, that means your prognosis is bad. So actually we can predict the prognosis because the ADC shows a very high correlation with the Gleason score. Okay, so these points are actually, each point represents a patient. So we collected many patients and we measure the diffusion image, and also they got biopsy, so they have their Gleason score, and then we do the correlation and get this result. So this is one application of the diffusion MRI. We can use diffusion MRI to detect cancer cell, and this is an example. We use it in the prostate cancer. And today, diffusion MRI can also be used to detect liver cancer or breast cancer. This is already being commonly used in the clinical applications now. And here I show you another application of the diffusion tensor imaging in the heart. This is the heart. I don't know whether you can know. This is the heart. Okay. This is actually my work back in 2000. And this is the first diffusion tensor imaging in the living heart. Very, very difficult, but we made it. And this, actually, the cylinders here are representing the fiber direction of the heart. So heart also is a muscle, right? So muscle are all fibers, right? So we can detect the direction of the muscles in the heart. So, and then we calculate how the heart fibers change their orientations from outside of the wall into the inside of the wall. And surprisingly, we found a very symmetrical orientation pattern. In the outside of the heart, the fibers are more or less this direction, okay? And in the mid wall of the heart, the fiber is more or less like this direction, circumferential direction. And then go further into the inner side of the heart, the fiber goes back to this direction, perpendicular. So actually, it turns from outside to inside, from vertical to horizontal to vertical. So it changes the variation. It varies the orientation by 180 degrees. Here, like this. 转了180度 from outside to inside. And this angular orientation is very symmetric. You can see, very symmetric. And I was shocked when I did this analysis. I found that heart is so simple and beautiful, like this. Okay?
So sorry. So heart is beating. So what's the typical delta time you use in MRI?
Okay. Very good question. This is a question from expert. Only expert can ask this kind of question. Because the heart is moving. So how can you control the big delta? Because the big delta is the time interval we allow the water molecules to diffuse, and then we detect the attenuation coefficient. But when heart is beating, how can we control the big delta? Actually, we use a technique called cardiac gating. Cardiac gating means we only detect at the same time point in the cardiac cycle. Okay, so heart is pumping periodically. So actually, it is like a period. So we only detect a particular phase in the period to detect the diffusion. So we can use this kind of a cardiac gating technique to get rid of this problem. Yeah. Okay. And then we start to analyze how the heart fiber is organized. So here, this is also diffusion tensor imaging. And we do a further step about the diffusion direction. Originally, the diffusion direction is all local, right? This is local direction of the fiber. And then we do a trick. We connect this local direction into the tracts, okay? So fiber tracts, that means tractography. We connect all the local fibers according to some criteria. For instance, it's like a streamlining. If you use some software, MATLAB, you will see the streamlining. Streamlining means if there's a vector field, you just connect the vectors that are very similar and have a small angular deviation, and you just connect them into curves, right? So this is called tractography. So we just use this kind of a tensor local first eigenvector map, and then we do the tractography, and we can get the fiber of the heart like this. So here is the fiber of the heart, over the heart. over the whole heart. And we found that actually there are two parts. One is the inner heart. This is the inner heart. And this is the outer heart. So hearts are organized into two systems. One is inner heart and one is outer heart. Why is that? Why is that? First, we look at the inner heart. So this is the inner heart, and this is the outer heart, okay? And why the heart has two parts? This is the inner heart. Actually, inner heart has two parts. One is like this. If we look at this fiber from top to down, it looks like this, like a tornado, So actually, the heart fiber is going this way. A part of the heart fiber in the inner heart is going this way. 有点像弹簧一样. And so when the fiber contracts, this will turn. There will be a torsion. If the heart the muscle contracts, there will be torsion. And also there are other inner heart. The fiber is perpendicular. So if the muscle is contracting, this will shorten the heart. Okay, so it's like heart is doing this action, torsion and shortening. Heart is doing this. The inner heart is doing this. So why the heart is doing this? Why? Squeeze the blood outside. Squeeze the blood and pump the blood with high pressure. So this is the action. Okay? So this is the inner heart. And this is the outer heart. When we look from top to down, the outer heart looks like this. Most of the fibers are like circular oriented, okay? So actually it's like our pidai belt. So when the muscle contracts, it works like this, right? So actually when the heart is contracting, it goes like this and this. All together. And that squeeze the cavity. This is the cavity of the heart. Squeeze the cavity and pump all the blood out of the heart. So this is how heart is working. So this is a very interesting study. We studied the organization of the heart muscle, and we found that there are two systems, and each system is responsible for a specific function of the pumping action. So this is the heart. And then we turn to the brain. Brain has another challenge. As you can see here, the fibers in the brain are very complicated. They actually crisscross to each other, right? So the diffusion tensor model no longer holds in the brain because diffusion tensor is the first approximation of the PDF, and in this area, actually the water molecular diffusion is more complicated than what the tensor can model. So here we have a challenge. So we actually can map the brain fibers like this. If we do correctly, the final result is like this. Can you appreciate the beautiful image of the brain? This is the conventional MRI. We can only see gray matter and white matter and CSF. But if we turn on the diffusion MRI, there are many abundance of great information inside the brain. And these are the fibers. These are all the fibers. So, but we need to use a technique that is more advanced than diffusion tensor imaging. And this technique is called diffusion spectrum imaging. So diffusion spectrum imaging basically is to measure the PDF, probability density function, rather than diffusion tensor. So actually we try to use diffusion spectrum imaging to measure the probability density function. Because if we have, we measure the probability density function, then we can determine the directions of the brain fibers. If there is crossing, we can still determine the direction of this fiber crossing, like this. So as compared to the tensor can only determine one single direction, and this direction is incorrect. But using diffusion spectrum imaging, because we measure the probability density function, and we can actually determine these fiber crossing directions. So this is the reason why we need to develop diffusion spectrum imaging to solve this problem, the fiber crossing problem. But there is a cost. The cost is, as you remember, in the DTI, diffusion tensor imaging, you can only you only need to turn on at least 6 diffusion gradient directions to solve for 6 unknown coefficients and determine the diffusion tensor. But for the diffusion spectrum imaging, we need to turn on hundreds of diffusion gradient directions. So the scan time is much longer than the diffusion tensor imaging. So this is the price. We need to pay this price in order to solve for fiber crossing. So actually, we implement this DSI sequence to the human application. So here, we apply more than 100 diffusion directions. And then we solve for probability density function. And then we calculate the orientation distribution function. And then we determine the local fiber directions at each position. So this is the flow chart of our DSI. I can tell you that we spent about 5 years to reduce the scan time. Originally, it takes about 3 hours, and it is not feasible for patients. And we use many acceleration speed techniques to speed up the DSI technique. And now we can, it takes about 15 minutes to complete the scanning. And we now do this routinely in our patients to get the diffusion spectrum imaging. So here, once we acquire the DSI and we can calculate the ODF, orientation distribution function at each position, and then we can determine the local fiber directions at each position, and then we can do the tractography to get the brain fiber like this. So this is the first application in our patients. Actually, this is the fiber tracts. This tract is the tract connecting from our eye to the visual cortex in the back of the brain. So this is the back side of the brain, okay? So here, the green fiber here is the optic tract. Do you know optic tract? 视觉神经, optic tract. And this is the patient who has a tumor over here. So this white region is the tumor. And you can see the tumor compresses the optic tract over this place. This place has a particular name. It's called geniculate.